So this is a talk on biomarkers on ADHD. I will cover some review papers, some meta-analytic papers about what has been found for biomarkers, but I will also spend quite some time on the principles of biomarkers. So a biomarker is something more than just having a biological assessment, an assessment of a biological measure. If you want to validate a biomarker for EMA or FDA, biomarker studies should follow the same principles of stringency as when you want to validate a new molecule, a new drug, a new medicine for clinical applications. That I will explain that later. And then at the very end, I will give some examples of biomarker research more going into depth where I'm involved in for autism. Just my conflict of interest. I have been and still am working with pharmaceutical companies for giving them advice on developing better medication. But of course, medication development is not the topic of this talk today. So I like to start with a successful approach from medicine. It's a paper not about ADHD, not about psychiatry even, but it's a paper about a successful biomarker approach. So it's a paper on drug-drug interactions in general medicine. And I saw in the TIMESPAN protocol, the TIMESPAN application, that there is also research here to which extent, for example, drug-drug interactions, medications for ADHD, but also medications for somatic comorbidities, type 2 diabetes, and so on, could interact. But this is a worldwide study. It was led by people from the Netherlands, not from Nijmegen. And they had a 12-gene pharmacogenetic panel to prevent adverse drug reactions. Open-label, multi-center controlled crossover implementation study. 18 hospitals around the world, community mental health centers, GP practices, and pharmacies in 7 European countries. Participants were genotyped for 50 variants in these 12 genes. And then, if on the basis of the medication to be prescribed and then a pharmacogenetic knowledge, there would be a signal that the drug prescribed either or the dosage of the drug to be prescribed had to be adapted. That was one arm, and the other arm was care as usual. So in half of the cases, they used the information from the pharmacogenetics panel to adjust the medication, and in the other half of the panel, there was no use of the genotyping information. Now, the outcome measure, the dependent measure, was the percentage of drug side effects. And the key, we won't go into the details about gatekeeper 1 and 2, but here you see the control group with the event rate of adverse effect of drug side effects. And this is the intervention group, and depending, irrespective of the gates, there was like a lower percentage of clinically relevant drug reactions, 21%, in the target group versus 27.7% in control group. Demonstrating that using genetic information as a complex biomarker panel could be clinically effective, lowering the chance for adverse drug reactions. Now, of course, is this fantastic? It was good enough to be published by Lancet. That's for one. Another question would be, is this cost effective? Because it's expensive, it's still the whole machinery, getting the genotyping, getting the algorithm. And you could also say, well, it's statistically significant, but it was a huge sample size, and lowering the chance from 27.7 to 21% is indeed, let's say 7, it's about 30% reduction, but it's not dramatically, it's still 21%. But still, this could be a wider application of using biomarkers in future in general medicine, maybe also in psychiatry, maybe also in ADHD drug medication work. And it could be that there is a future that, for example, we use genetic information to adjust dosages of stimulants, Ritalin, or other ADHD medication. Okay, let's starting with looking at some definitions of biomarkers. Biomarkers are measures that characterize baseline state, a disease process, or a response to treatment. They can be physiological state, pharmacological responses, disease characteristics. And changes in biomarkers following treatment reflect the biological response to the product. This is a definition that a little bit tries already to link the biomarker to go further than just a biological measure. It's a biological measure that to some extent has a link with the pathophysiology involving the disorder, or the pathophysiological process, or the pharmacokinetic process of the medication response or the drug response. This definition is more neutral. It is a measurement that suggests etiology, susceptibility to activity or progress of the disease. It's a little bit more neutral. And this is even more neutral. It is a biomarker that assesses different biological characteristics. Now, this definition is not very informative. So I like to argue that a biomarker that's useful and that's to be implemented should have a link with an underlying biological model, should be biologically plausible, and or should be linked to the prognosis or the prediction to the response, and should be more than just a biological measure. This is one way to classify biomarkers. So, as one of you already said, it could be you have a diagnostic purpose. You use the biomarker to predict classification, DSM-5, ICD-11. And this can be used in the clinical and in the research tool. You can use it as, okay, I shut off my clinical brain. I ask the biomarker, does this patient has ADHD, yes or no? And blindly, I follow the output of the biomarker algorithm. Will that ever happen? The way I phrase this is unlikely. This will not happen. But it could be that you build it in a decision support tool. But at least this is a diagnostic marker, and this diagnostic marker, of course, would meet criticism for the whole DSM, ICD-10. In fact, the criticism for whole psychiatry, that many criticisms of psychiatric classification say all these diagnoses are just subjective. Every diagnosis, also the ADHD diagnosis, we don't have gold standard measures, clinical, cognitive. It's all questionnaires, it's all behavioral assessments, and it's some vagueness. What is clinically relevant in attention? Now, of course, as a clinician, we can talk long about that, but this tries to overcome that. Now, then we have risk markers. Risk markers are used for detecting the disorder, the problem, the disease earlier, can also be used to stratify. So, for example, you have preschoolers or you have very young infants. They have a little bit difficult temperament. They are a little bit active. But every preschooler is, of course, more active, more hyperactive, shows more motor activities, more impulsive than older children. And the key point then is, can we predict those preschoolers that will further develop full clinical ADHD? Can we differentiate them from those maybe high risk who will not go onto the clinical pathway? That would be a risk biomarker. And then this could be a potential strategy to start prevention programs. Prognostic markers are markers that give you a prediction about the course of the disorder. So, for example, we have a 15-year-old or a 17-year-old adolescent with ADHD, and then the adolescent, him or herself, but also the parents, the teachers, how likely is this to be persisting ADHD, to be a persistent adult form of ADHD that will be long-lasting over adult, middle adult ages, even old ages. So you will predict the course of the disorder. That's a prognostic biomarker, also from preschool ADHD to childhood ADHD, but particularly from adolescence to adulthood. And this biomarker can be used to differentiate between persistent and remitted course of ADHD. Then the prediction biomarker is the biomarker that predicts the response to treatment. Is this a form of ADHD, an individual with ADHD that's particularly better responsive to, let's say, stimulant than to non-stimulant medication? Or this a form of ADHD that's more responsive to behavioral therapy, or modern lifestyle, diet, neurofeedback, meditation, mindfulness, swimming with dolphins? No, that's not evidence-based, so forget it. And then, very important, and in my view, my personal interest is mostly in this: can we use stratification biomarkers? So all ADHD, all psychiatric disorders, all autism are extremely heterogeneous. So they're probably based on different, not one, but it could be 3, 4, it could be several underlying pathophysiological pathways, and then a biological subtyping or a stratification marker that subtypes into biologically and or clinically more homogeneous subgroups could pave the way to precision or personalized medication. That's the stratification marker. The biomarker, by definition, tells you something about the individual. So if you start biomarker research, you should completely drop the old case control paradigm. 99% of all papers on ADHD give you case control comparisons, effect sizes, standardized mean differences, p-scores, p-values about how the cases and controls differ. That tells you about the group, that on average, the average ADHD individual in the ADHD group is different on average from the typically or control individual, the non-ADHD individual, but doesn't tell you anything about the individual prediction. So when talking about biomarkers, we have to go to individualized statistics. So this is an example, heterogeneity, and we have different individuals with different biological, genetic, other characteristics. And we may think about, and of course, mostly we take the example of medication. We can adjust this in terms of type of medication, dosage, duration, administration route, and so on and so forth. This is a paper we developed in our autism consortia on the significance of the mean group differences for biomarker discovery. In typical statistics, in Cohen's D, for example, an effect size of 0.2 is a small effect, small case control difference. An effect size of 0.5 is moderate, and an effect size of 1.0 is even the effect size of stimulants, the most powerful medications in psychiatry. But even an effect size of 1.0 gives you only 16% difference between the controls and the cases. So 84% of the cases and controls overlap for that characteristic. So even if you have a fantastic finding, a huge group-based case control difference, it will never give you a handle on, let's say, if you would like to separate the two groups, because 80%, 84% overlap between the cases and the controls. So even to separate the two groups for about 75%, you need an effect size of larger than 2, and to separate the two groups for about 80%, you know, most, let's say, a common sense kind of criterion for diagnostic accuracy, if you like to go for a diagnostic marker, is at least an area under the curve of 0.80 for 80% accuracy. So you need, if you would go for one measure, you need case control differences from that measure with an effect size 3.0 or larger. Now, I told you that it's far more work than just to say I have a nice study. And so going into the biomarker route, and also if you like to go for clinical qualification, that's the technical term where EMA or FDA would accept your biomarker proposal to be qualified, like they can give you a license for a medication, you need through all these different stages. Let's say you go to EMA, and you go to EMA not with, oh, let's drink a cup of coffee. You go to EMA with already a very much pre-planned idea of your study protocol. You go to EMA and say, okay, I like to do a study with this and this samples, and I have a number of candidate biomarkers. For example, event-related potential measures, eye tracking for autism, MRI, serotonin, mention it. And then EMA says, okay, how can you justify that? What's your idea? So it's like when you go for a medication study, you have phase 1 and phase 2 studies. You have already data in the pocket that suggests your biomarker to be somewhat plausible, that it's likely that our understanding of the pathophysiology or the treatment response has a potential stratification. So you develop a protocol also with the clinical measures, and then you get discussions with EMA or the FDA, and you already get discussions about this COU. Remember, it's very important. Context of use. So it's not a biomarker that you can say, oh, maybe tomorrow we have an idea. So you should from the very beginning already think about, is it a diagnostic marker? Is it a stratification marker? Is it an enrichment marker? That's another way to let's say, to enrich your sample that you recruit for a clinical study, for example, for certain characteristics. It could be a safety marker. It could be, I didn't tell about it, a surrogate endpoint. So, for example, suppose you work on a biomarker that aims to prevent the development of dementia. Yeah, but the development of dementia is a process of many years. So you would then have to run a clinical study for 5 to 10 years. But if you can say, I have an intermediate biological process, let's say accumulation of amyloid, which is maybe questionable for the experts on Alzheimer's disease, but you may have an intermediate endpoint and then say, if I reduce the amyloid substances in the brain, that's the predictor for later Alzheimer, and that's then the context of use. So you start to talk with EMA, and then they give you advice, and then you revise your protocol, and then when you get your approval, you go to the ethics committee, you check your funding, and you start a study. Then there is pre-analytical validation, analytical validation, qualification, the submission of regulatory approval. So this is very boring stuff, of course. I'm aware of that, but it's not too late today. But you have to check, is the biomarker easy measurable? What are the test characteristics? So if it's eye tracking, is the calibration easy? Is the calibration similar in different eye tracking labs? The lighting conditions, the computer, the software, you have different types of eye trackers. Can it be standardized? What are the standardized operating procedures? Quality assessment and quality control issues are very important. We think this is all important for laboratory studies, so fluid biomarkers, blood or plasma biomarkers, but also for EEG, ERP, eye tracking. All these standardizations are important. Then you have to think about analytical validation. What's the sensitivity, specificity of the marker? The robustness. Is there a reference score? Can you individualize the score? Then for the qualification, that's the most difficult. You have to demonstrate a link with a significant relevant clinical endpoint or measure. That could be outcome. In oncology is of course mostly remission of cancer. In ADHD it can be also remission, but can also be no comorbidity, or can also be adequate response without side effects. adequate response and well-being, higher rate of well-being, for example. So there should be a clinical endpoint, and you have to go through these processes, and then you can submit to EMA. Now, this involves the pre-analytical validation. So the test characteristics involve a lot of work, because most of the time this is multi-site studies. Some measures are more easy to be standardized over different analytical sites, different labs than others. From my experience, for example, you could say EEG or ERP studies were very difficult to standardize, because there is different methods for, let's say, quality control, outliers, movement detection, movement artifacts. This is all improving. We improve in developing better algorithms to qualify, to quality control the data, and so on and so forth. But this should not be neglected. The analytical validation is the accuracy. So that's the difference between the cases and the controls. It's a diagnostic marker, but it's also the precision. So it's the measurement error. How large is the measurement error compared to, for example, the effect size or the odds ratio or the power, the robustness of the finding? Can different findings be reproduced by other sites? If they have, let's say, different findings at different ages, for example, is that reproducible at other samples in other sites? Is there reference values? And can you harmonize the analytical performance? Suppose you have a fantastic finding and you are in a consortium with 3 to 4 other sites, which is quite usual now. And if you have drug medication studies, you may have even 20 sites. And only 2 sites have a fantastic finding and the other sites cannot replicate this. You are in a deadlock. You will never come at a qualified, accepted biomarker. But then the interpretation, the post-analytical validation, the interpretation, it's like, of course, sensitivity, specificity, but also like, if you have a continuous biomarker, what is the clinical cutoff, given the biological hypothesis, the biological plausibility, that has to be agreed upon between the different sites. The reliability, and with data-driven subgroups, I come to the topic later for stratification, and is it also reproducible? This from the FDA schedule. You can have 3 different stages. You can have an exploratory biomarker. That's where you start. You can have a probable. If you go to EMA or FDA, you should at least have a probable biomarker, because you have some pilot data and you have some idea where you are going to. You see the future for you, like this can be potentially useful. And you have a known biomarker. And then you are already in the stage of further replication in independent cohorts, different ages, males and females, diseases, ADHD with and without comorbidity, non-ADHD controls, or other disorders that are not typically developing or healthy controls, but controls with other disorders, and so on and so forth. That's the known valid biomarker. Now, let's give you some examples. There is a lot of efforts in finding MRI-based diagnostic biomarkers. I will not go into the individual underlying studies. 55 studies were meta-analyzed and reviewed in this recent paper. It combines diagnostic algorithms, machine learning approaches to either structural MR, cortical surface area, cortical thickness, overall gray and white matter issues, different features. You can, of course, if you go for cortical thickness, you can go for cortical thickness in 20, 40 different cortical fields. It's numerous different variables, functionals, resting state or task related, is all together with different statistical approaches, and more classical machine learning support vastly convolutional neural networks are deep learning approaches, other statistical approaches. It's very important how you select your test sample and how you select your training sample. So whether it's balanced versus unbalanced. Balanced means that, so if you try to develop a diagnostic marker and your test sample or your training sample has 50% ADHD cases, 50% non-ADHD cases, is balanced. If your test sample has 80% ADHD cases and 20% non-ADHD, your average statistical maneuver is biased because the chance that you will detect a priori ADHD is much higher than the chance that you will predict a priori non-ADHD. So you have to control for that, and it's more difficult. And particularly also in the final test sample. And so some studies have a balance, some have unbalanced. And then it's very important to evaluate model performance and generalizability. Do you really try to replicate it in a completely independent sample, is the hold-out test, or if you have some cross-validation left out, or k-fold cross-validation. Just to explain this is the cross-validation where you have a training set, and typically this is done, I thought also in the work package in TIMESPAN, I think it's written by Steve Varone, that he has like 70%, or mostly you have 70 or 80% training, and then you have 20 or 30% validation set, but at least these are completely independent. So you have training, or you may have discovery, you start exploratory work, then you have a basic algorithm, then you optimize the algorithm in the training set, and then in an independent test or validation set, you check it again. This is one way. All the ways are leave one out. So you have the whole set, and then every time run through the statistics, and every time you leave out one sample or one case or part of the data set. That's this approach. And this is the approach when you have k-fold. So you multiple times run through subdividing the whole data set in folds, and then you check whether you have a stable output, and you can estimate the confidence intervals. This gives you the more stringent data, the better data, and I will show that this has an impact on your overall findings, because this is from the same review paper, the accuracy by test type. So if you do the held out test, accuracy 80%, perfect, 100%, you see that overall accuracy is lower and probably more just, more fair, more realistic if you do the held out test. And if you do leave one out or the k-fold cross-validation, you may overestimate accuracy. There's always an issue like we become, or statistics becomes better, or sample sizes become larger over the years. So is there a link with the years of the study? You see that for the k-fold and the leaving one out, you see here slightly that you have improved accuracy over studies published later. Is also the case for the hold out test. But you see that, so the validation procedure is very critical, and so you can easily overestimate the accuracy of your test if you have a k-fold or leaving one out approach. This is the effect of sample size. Overall, larger sample sizes, particularly with the hold out test, give you lower accuracy. Now the question is, why? What's the explanation that if you have a larger sample size, your accuracy is lower? You would expect, like in classical statistics, which have better sample size, you have more power. You can do more iterations, you have more power, you are in business. And now we see it's quite the opposite. Now, the likely explanation is larger sample sizes are more real-world data sets. Real-world data sets meaning with all the noise, statistical noise, measurement noise, but also heterogeneity. I can tell you with Catharina from our starting point, so around 2008 in the Netherlands, we got a grant for building a very large MRI database on ADHD. It's called a neuroimage study. We had a sample size of 800 scans, about 300 ADHD probants, about 200 controls, and then we had also affected, unaffected siblings. Before we did that study, the typical sample size for MRI studies in ADHD was 20 versus 20, 30 versus 30. We said, we have hundreds. We will solve every ADHD problem. We thought we have such a fantastic sample size. Our power is great. But what happened? Our case control differences were smaller than expected overall, not for all measures. And that's because we realized we sample more heterogeneity within group variation, and you see case control differences. If the within group variation is larger, and then you test for the proportion between the between group variance and the within group variance, it's more difficult to find case control differences. And we had to acknowledge the ADHD brain is much more similar to the non-ADHD brain, and then it's different from the non-ADHD brain. The same applies for depression, the same applies for schizophrenia, the same applies for autism. So overall, MRI diagnostic models are not good enough for clinical application. Sample size is one major bottle. No implications for whether you use structural or functional MR studies. Most studies are on children, and the validation method really matters. That's the conclusion. Now, let's reflect somewhat more on this MRI approach to diagnostic markers. Suppose you have a study or you have an algorithm and you have opportunity to have MRI scanning. And the algorithm tells you the probability that your patient has ADHD, that your patient has an ADHD brain, is 70%. And then, what's then? How are you happy as a clinician? Are you unhappy as a researcher? Are you happy? Are you unhappy? What does that tell you? Now, let's say the clinical presentation is atypical. It's not a typical, like the presentation does not typically satisfy the DSM criteria for ADHD. But still 70% is the brain likely to be the brain of an ADHD person. Would that make your task easier to say, I try to check for other diagnostic issues? Or would you say, well, 70% is more than 50? It's not 80, but it's more than 50. So in this case, if you would say that the presentation is very typical, so it's really fitting the DSM algorithm, but you say 70%. What I like to argue is, in my view, these MRI studies are not so helpful. They may be even confusing, also at the individual level. So, case control differences are generally small. The ADHD brain is more similar than different. And another problem with all these, but that is also for EEG algorithms, for other algorithms. The algorithm doesn't tell you anything about the pathophysiology, because the algorithm is a black box. So if you would do a regression, let's say you would do classic multivariate analysis. You have a number of predictors, cortical thickness, whole brain volume, white matter, gray matter. And you would do classical regression analysis, logistic or something else, and then you would try to predict ADHD versus non. You can see at each individual measure whether they contribute or not, and that could help you seeing, I see is more stronger contribution by the white matter variation than by the gray matter. So probably white matter tracks are more important for differentiating between the cases and the controls. That's, for example, what we found in our autism studies, that white matter tracks are stronger impacting on case control differences than gray matter. But the machine learning algorithm gives you the output, the statistics, without giving you insight into the underlying process and the underlying variables that are included. And then one way is normative modeling. Just one way, a few words about normative modeling. The whole issue is that this allows you to move away from group-based statistics to individual statistics. So So this is the typical case. You have cases and controls. You have even different types of cases. This is the heterogeneity. But the question is whether this is the more realistic situation, where cases and controls are not from two completely different populations, but are examples from the same population, and cases and controls are not easily to be demarcated and are part of the same biological distribution, or at least you need a complex multivariate approach to separate cases from controls. Now, in the normative modeling, your approach to a biological measure is the same like growth charts in pediatrics. In pediatrics, we have a growth chart for weight and height, and for girls and boys. And for weight and height, we can, per individual, we can tell you, okay, your child is for his length at a P90, means he is among the 10% tallest for his age. You give an individual data point, you give an individual description, you quantify the individual variation. Now, you can do, in fact, the same for all biological measures. You build a normative model. You have to start with a large population of normal individuals. And for example, you build a normative model on cortical thickness, on hippocampus volume, on cortical surface area, on white matter tracts, on an activation to an inhibition task. You build a normative model. Mostly you build in age and sex. You can build in, if that's relevant, SES. And then you can tell, this is the next slide, you can quantify the uncertainty of your measure, but you can also quantify whether that child has, for his age and his gender, a cortical thickness at some areas that's in the bottom 25% or is average. That allows you to make better predictions at the individual level. My colleagues in Nijmegen, Christian Beckman and André McCann, are really expert on this, and so typically they are now improving their methods. They work on non-linear methods, probabilistic methods for different ages, and making the model much more complex. And it's now widely used in all big data sets. But you quantify the individual variance at the individual level. You remove the need to artificially dichotomize into cases and controls. And pathology is seen as redefined as a deviation from the normative model. Now, quickly, some other diagnostic, there's another review paper that also reviewed MRI studies, but also EEG, MEG, and FNIR studies for diagnostic markers. Here, the same, like only slightly more than 50% used independent data sets for replication. Non-independent data sets, 50%, no cross-validation, not reported. Different statistical methods, but this is the bottom line, that independent data sets are insufficiently used. Here you see the same, that if you have larger independent data sets, you lower the inflation, you have a more acute measure, but your accuracy is lower. If you have non-independent data sets, you overestimate, and here also greater variance in the later. Just to give you some other examples of potentially candidate biomarkers in the ERP, event-related potential research for ADHD, you know, the early waves of the event-related potential, the P1 and the P2, are more reflective of early sensory processes. The P3 or P300 is reflecting the higher order cognitive processing of stimuli. If you look at the meta-analysis, you see that you have all significant case control differences, shorter latencies to the P1 or P100, smaller amplitudes to the P300, longer latency to the P300, so the later cognitive appraisal of the stimuli, cognitive elaboration. And these group differences are moderate, so meta-analytically robust, confirmed. But you see, this is far away from useful for clinical test and clinical diagnostic applications. This is just to show you the meta-analytic evidence for this. Some years ago, there was some hype about could it be easy, like you quantify the proportion of theta waves in the EEG and beta waves of the theta power and the beta power. The theta waves are slower waves, 4 to 7 hertz. The beta waves are signs of more brain activation, faster waves between 12 and 30. And so the proportion of proportionally more slower waves and fewer fast waves would be indicative, or let's say a somewhat sleeping brain, a low aroused sleeping brain. That would be the ADHD brain. Now, very interesting. Earlier studies found that to be diagnostically useful, found a higher ratio, but then over the years, studies declined. Studies found less significant findings for the theta-beta ratio and not any use. It's very interesting. Has the brain of people been changed over the time? Or maybe we did select brains in a different way in early studies. Nobody knows. I just published another review on diagnostic markers. One of the studies included was the continuous performance task with movement analysis, the QB test. The advantage of this test is that it tests for, is a cognitive test for impulsivity and inattention. The impulsivity measures are overly responsive to non-cues, and the inattention measures are not responding enough to cues. So it has a operationalization, cognitive operationalization of inattention and inhibition and impulsivity. And then there is infrared camera, and it takes your movement while performing on the task. So it has measures for all the 3 core components of ADHD. So at face validity is high because it's no hocus pocus. It's not very complex. It's very directly measuring, objectifying the key features. Well, 11 studies, case control design, sample sizes overall much too small. Areas under the curve range from 0.5 to 0.80. This is quite good, but not anyone did out of sample testing, overinflation, and it's too early to see this as a diagnostic test. It could be helpful for diagnostic support system. And a very interesting study, I like to flag this study, because this was a study by our colleagues from the UK, Chris Hollis, and he did a diagnostic trial. So what he did is he had an outpatient clinic, and he ran a diagnostic RCT. And so he randomized potential ADHD referrals across an arm where the QB test results were disclosed to the diagnostic team, open. And for the other arm, the QB test results were kept hidden. And what he found out is that in the open arm, there was a faster diagnostic decision compared to the blind arm. This is an example of a diagnostic support system. If you feed in clinical information, interviewing, questionnaires, developmental history, and you add in relevant cognitive information, I think this type of diagnostic RCTs merit more attention. Now let's go to predictive markers. What are predictive markers that can help us making a treatment decision or can be helpful for monitoring treatment effects? Fantastic review by Michelini from, she's now in the United States, but she was involved in King's College in London. To make a long story short, the design of these studies that were reviewed is this. You have a sample of ADHD participants. You have baseline information either from MRI, structural, functional, resting state, or EEG or AP or genetics. Then you have either in an RCT or in observational design, you establish whether there is treatment response or non-response, and then you check whether responders, non-responders differ in earlier baseline characteristics, and whether earlier baseline characteristics like certain MRI or EEG or genetic features could predict treatment response. Well, overall, there were some nice studies like different activations of the caudate nucleus predicting stimulant methylphenidate response, but a worse response to atomoxetine. There were some studies suggesting that higher theta power during the resting state did predict response to stimulants, to methylphenidate. But the prediction from theta-beta ratio was inconsistent. And there was also one other study where they were comparing methylphenidate, guanfacine, and non-stimulant medication for ADHD and a combination. And in that study, EEG measures explained twice as much variance in response than did clinical measures. So this review has massive amount of tables with all the details. But the bottom line is, if you would go for a predictor, the EEG probably, the spectrum of the EEG, theta, is probably better, give you more chance on a predictive marker than MRI. Then they did also review whether you could have monitoring markers. So for example, if the treatment would induce a change in a certain biomarker, could that be seen as, oh, this is a clinical response? That could be useful. So I had also the experience early on. You start medication treatment. After 2 weeks, 3 weeks, parents say, fantastic medication. What you wanted, that they would say, you are a fantastic doctor. But they said it's fantastic medication. But then after 6 weeks, they said, I don't know. He did so well 2 weeks ago. But now we have some doubt whether the medication is really helpful. And that can happen. Changes in stress, changes in school rhythm, another teacher, bullied at school. But so there could be a case that you want an objective marker of the clinical response, and that could be done a biological marker. Now, for this also some relevant findings, but this is a very difficult area. It's not so useful for the moment. The conclusion from this review is not very positive that MRI or EEG, ERP measures can be used for monitoring markers. Cognitive markers probably are more useful. So this is a meta-analysis review from the effects on cognition from pharmacological treatment, cognitive training, and cognitive training where there was unblinding, so this could be the bias. Overall, many treatments, not only the pharmacological but also the cognitive training, did improve on some cognitive markers. So cognitive markers could be more useful, in fact, for monitoring treatment success and response than EEG or MRI. But of course, overall, these are the clinical markers, and you see that medication has the strongest impact on the clinical symptoms. What they concluded from this review is that the traditional cognitive model of ADHD would predict that if you impact on cognition, particularly cognitive training, so ADHD has been associated with deficits in cognitive functioning, inhibition, inattention, and others. If you would improve basic cognitive measures, that would be a mediator for improving on cognitive symptoms, on ADHD symptoms, and would lead to less impairment, better quality of life. Say this is probably not the case. If you work with cognitive measures, you have to see them as a parallel track. Some interventions have a stronger impact on the clinical symptoms. Other interventions have a stronger impact, or on another individual, on the cognitive measures. And the mediation model is probably not very supported by the data. Prognostic markers. So again, prognostic markers are markers that help you to predict the course. For example, in high-risk preschoolers or in high-risk adolescents. Or one example from a great meta-analysis for prognostic markers. This meta-analysis is built on reports on 2 systematic reviews and meta-analysis. One is on early signs and symptoms, early markers in preschoolers or very young infants with a familiar loading for ADHD, so they are genetically at higher likelihood for developing ADHD. And the question then is, do we have useful early markers that can predict whether they will transit to full clinical ADHD or not? And then are early interventions helpful? Now, here you see all these differences: cognitive ability, language. So those with high likelihood for ADHD at preschooler, they have generally lower IQ, lower language skills, less developed motor skills. The problem is they did not, you see, this is a negative sign. This is a positive sign. Would have been better if they would have rescaled all these effect sizes with the same signal, same sign, plus or not. But they have more problems in social functioning, more problems in emotional functioning, more sleep problems. This is a whole bunch of, yes, at the group level, high-risk preschoolers. At the group level, they have multiple group level differences between non-ADHD, low likelihood ADHD preschoolers, but it doesn't tell you about the individual prediction. And so, of course, clinically, you can still use this information, but it doesn't tell you. You cannot give, say, this is an 80% chance that your child will develop into clinical ADHD. I would even say, but I know Catharina, with some of my colleagues in the Netherlands, is running such preventative studies in the Netherlands. It could even be harmful, of course, stigmatizing, and that the whole biomarker issue has some ethics. It's not on my slide, but we can discuss this at the end. But it could be even stigmatizing if you would say to parents, oh, your child has 80% chance to develop ADHD if you have a 4-year-old who's a little bit distractible and hyperactive. It's maybe much better to say you have a hyperactive, your child is very motorically active, and maybe we can help you handling your child. So a little bit could be useful to stay away from diagnostic classifications and diagnostic statements early on to prevent potential stigmatization. And for some parents, it may be helpful to know, but some other parents may even get a little bit demoralized, say, oh, if my child will develop ADHD anyway, what can I do? Not terrible. Nothing terrible. It's just what can happen. For the interventions, they also tested for behavioral interventions. And then these early interventions, preventative interventions, were more powerful in improving the ADHD symptoms than they were powerful in improving the associated symptoms. Then is genetics any useful? One way to look at genetics for diagnostic markers is to construct polygenic risk scores. Probably you know that, maybe better than I know. So you have a large case control data set. You look at all the SNPs that are statistically different between cases and controls. You weight them by their effect size, and that allows you to construct a polygenic risk score that's in principle normally distributed. And then you apply the polygenic risk score to another cohort and then give you the polygenic risk score. And then you have, of course, high risk and low risk scores. Now, diagnostically, this is from the latest GWAS report that's published. I know there's another report in preparation or maybe already submitted or accepted already, but not published, I think. Oh, okay. At least this is done the pre-last, and it shows you that the higher polygenic risk score, if you are the 10th percentile, your odds ratio of having ADHD is 5, but it's still 5. It's a high odds ratio, but it doesn't give you 80% accuracy prediction. From the ICPSR data and from the PGC data, it's even lower. So the polygenic risk score gives you some diagnostic probability, but not for a diagnostic test. The polygenic risk score is not that bad for ADHD. It's for research purposes very useful because it differentiates between remitted and persistent cases. The higher score for persistent cases compared to remitted cases and non-ADHD. And it did not differentiate between the comorbidity for ADHD or alcohol use or marijuana use. So the polygenic risk score is potentially maybe something to be further developed if you have better GWAS data, and could be useful for still indeed decision support systems, but in terms of probability, not in terms of having a final accuracy measure. The polygenic risk score also, so it did correlate with the ADHD diagnosis, ADHD trait. It did correlate with the number of brain measures, educational performance, other externalizing behaviors, general mental health, neurological test performance. Overall, not too bad, not too bad. Interesting, I would say, for research, not for clinical application. The stratification markers. The stratification markers is, put the ADHD also in the broader perspective of the comorbidity with other neurodevelopmental disorders, like we have ADHD, we have overlap with autism, we have overlap with intellectual disability, we have overlap with motor problems, and so on and so forth. And so, can we subtype this, either the ADHD or the broader group of neurodevelopmental disorders, into more biologically homogeneous subtypes? Well, the reason is, of course, heterogeneity, clinical, severity, spread, comorbidity, different scores over different symptom domains. We have etiological comorbidity. For the polygenic risk score, the score is only relevant for common genes, but we have also rare genes, not quite crystal clear to which extent copy number variations or rare mutations or strong genes with a major strong effect account for ADHD. Could be 5 to 10% of ADHD. It's not that clear. And then, of course, still there is a lot of research needed for a better understanding of to which extent also environmental factors, together with genetic factors, predispose to ADHD. To which extent early trauma, maltreatment, abuse and neglect. It could be other environmental factors, biological environmental factors, pollution. The more we know about the genes, the less we know about, we discover the less we know about the environment. Our hope is on people like Katarina, who run very big cohort studies, epidemiological studies, who should clarify the role of environmental factors. And then we have, of course, biological variation. I already told that probably all cognitive and brain systems could be implicated, but at a deeper level, probably there could be several major molecular or biological pathways driving ADHD. And then, why is this important? Well, this is in fact what we know from botany, from flora and fauna. If this would be all orchids, we know some orchids need a little bit water. Some need more water. Some need a ground with a lot of chalk. Some don't like that. So biological variation is related to flourishing in a specific environment. And environment is also treatment environment. So this is in fact the principle. So suppose we have a heterogeneous disorder, ADHD. The one-size-fits-all treatment would lead to some effect of the intervention in a subgroup, no effect in another group, and even worse effects, adverse effects, so detrimental effects in other groups. So overall, it will be difficult to establish an overall group effect. And the idea of a personalized or precision treatment is you have a biomarker, could be a genotype, could be cognition, could be MRI, could be something else, and you give evidence-based stratification, and you stratify your treatments according to the biological measures, and everyone is happy. Now, this is a long way to go. This is a long way to go. But this is the way we should go. It's as simple. So, and how can we make efforts to go to that way? We have two strategies to subtype. As always, data-driven, hypothesis-driven. And, well, they are both useful. They're both useful, but they are most useful in combination. Just hypothesis-driven, good. We need more hypothesis, but also data-driven is also important. And then to try to mix and to combine whether they converge on certain solid subtyping. So hypothesis-driven is, for example, this. This is an example we use in some of our projects, that we say we have a number of risk factors like pre-perinatal, prematurity, low birth weight, genetics, neuroinflammation, and also the timing. And for example, in this paradigm, you would say that you expect, for example, neurobiological impact, for example, neuroinflammation, for example, infections during pregnancy, which may give rise to early maternal immune activation, which may interfere with cytokines from the maternal circulation coming into the fetal circulation and impacting on the fetal brain. And overall, you may suggest that the earlier these early biological factors are operating, the earlier the risk for autism or intellectual disability even, and the later they are operating, they may be impacting on the later risk for ADHD. This is a little bit theoretically driven approach. Another theoretical top-down approach is to say, and of course this is not completely theoretical because it's driven by preclinical data, that the balance between its excitatory inhibitory systems in the brain, the EI imbalance, is of crucial impact on the formation and the development of cortical systems early on. And so a disbalance between excitatory, that's glutamatergic, and inhibitory GABAergic systems may impact on early brain development and brain specialization, so cortical areas may become disturbed. That would impact on the risk for autism, ADHD, intellectual disability, and so on. So this is a little bit overarching top-down hypothesis-driven. Again, even if you do it bottom-up, data-driven, you can start with the brain and you can start with the symptoms. And again, you can argue for both approaches. You can say, let's start with subtyping at the brain level, circuits or systems, and then check for symptoms. Or you can start with symptoms and then go whether these subtypes differ on the brain level. This is somewhat the approach also advocated by Joel Nick, one of our colleagues from the United States, in a paper in Biological Psychiatry, where he says you can start with behavior and then map onto cognition and brain differences. You can start with cognition and brain and then check for behavioral differences. Or you can also work with the both together in already integrated analysis from scratch on. There is a lot of method sophistication about this approach, this stratification approach. I take this from a review from one of our colleagues, Hilde Geurts from Amsterdam. She's one of the senior authors. Because you could say how to subtype. It's the same like with the diagnostic marker, and then we talked about the machine learning and hold-out test or cross-validation. Even if you subtype, you have different clustering approaches like hierarchicking, k-means, you have latent class analysis, you have latent profile analysis. The message here is you feel more safe if different methods are converging on more or less the same subtypings. If a certain subtyping approach gives you a stable solution, but it cannot be replicated by another statistical method, you have a little bit to think about, okay, it's nice, but is it solid? Is it robust? Does it really taps into the really underlying biological differences? And do I have a core bottom of a biological system or a biological hypothesis? And do I get the same subtypes by different clustering methods? Is it replicable in different samples? Here again, the out-of-sample test. Is it stable over time? Is a subtype 2 years later, if I follow up, is it replicated? How sensitive is it to the original measures? Should I exactly use the same questionnaire? Should I exactly use the same processing of the MRI data or EEG data? Or can it be replicated by equivalent measures? If it can be replicated by equivalent measures, you feel more solid. You think, okay, it's not like this is the magic CBCL or SWAN or SDQ or for the ADHD. It's robust and it's replicable across different methods. And again, does it predict clinical validity? Does it predict clinical measures? Now, some examples. This was one of the earlier examples by Damien Fair on cognitive subtyping of a large sample of ADHD. They used community detection techniques that's based on the graph theory. We'll not go into that, but they used inhibition measures, working memory, arousal, response variability, different measures for each of these domains. And they were able to subtype the controls and ADHD into 4 profiles with differences between the different cognitive domains. For example, profile 1 had a peak in response variability. There was a dip in response variability. But the most interesting of this is that the subtypes in ADHD were very closely matching the subtypes in the controls. So what this study learned us early on is that we had also to abandon the idea that ADHD was heterogeneous and control was homogeneous. We have in fact also subtypes in the healthy controls. And so this conclusion of this paper was that heterogeneity in the disease, in the disorder, was rooted in heterogeneity in the population. Very important conclusion. Jeanette Mostert from Barbara Franke's group in Radboud did kind of the same approach in adults with ADHD, and she also found 3 subtypes, one impaired on inattention and inhibition, one impaired on delayed discounting, one impaired on fluency. And again, overall, she didn't use community detection, I think, some kind of cluster analysis, I think, but at least also more or less similar profiles, similar subtypes in the controls and in the ADHD cases. Another way to subtype, same. What all these studies did lack is clinical validation. So the proof of the pudding is these subtypes by Fair, by Barbara, would say subtype 1 is more likely to be persistent ADHD than subtype 2, for example. Or subtype 1 is more likely to develop massive substance use in adolescence than subtype 2. That would be a relevant clinical measure to say it's more than a statistical exercise. It gives us a clinical validation. Joel Nick had another way to subtype into temperamental emotion dysregulation or temperamental subtypes. For example, he made a case for having subtype low reward sensitivity, high threat sensitivity, so anxious and probably a little bit low aroused. The other way around, low threat sensitivity and high reward sensitivity, reward driven, and a regular group, for example, which is still a way to consider because it's more clinical features on ADHD than just the symptoms. The temperamental aspects and emotion dysregulation tends to be an important clinical feature of the whole clinical presentation. Then again, theoretically, could be like if the immune profile driven subtype is some part of ADHD driven by immune dysfunctions, linked with allergies, immune diseases in the family, autoimmune antibodies, both for autism and ADHD, for example, and a link with inflammation at later ages needs to be tested. We did a study on behavioral subtyping and then testing for cognitive differences some time ago, also with Katharina and others in Nijmegen. So we took a large sample of typically developing children, those with autism, ADHD in a combination. We subtyped them on behavioral measures, and then we checked whether they differed in some cognitive tests. And here you see that we also predicted that they would be more cognitively impaired when they would move from, let's say, typically developing controls to ADHD to autism, and the comorbidity would be mostly impaired. These are Z scores. And here you see that, for example, speed and accuracy for identification of facial emotions were more impaired in the combined subgroup. This was also found for attention measures. But for central coherence, for example, detail-focused cognitive style, we found that those with the combination of ADHD and autism, there was a subgroup which was very impaired, but another group who performed rather well. So this gives us a clue that, and we could replicate that in a population sample, that in those with autism and ADHD features altogether, there was a split in those with rather good and rather weak visuospatial skills. I have to say, we didn't check for clinical validity, which might have been done. Now, then of course, like the overall picture is to try to get subtyping that integrates along different measures, integrating genetic measures, physiology, inflammation, arousal, e.g., into different clusters. I have still 2 data sets to be presented. This is just one example of statistically top performance by my colleagues in Nijmegen, Professor Beckman and his team, because in a large autism data set, we did subtyping approaches using different measures: language delay, eye tracking, file length is an interview for social, emotional, and communicative performance. The N170 is a response on the ERP to faces, sensory profile, another measure of sensory profile, and cortical thickness. So we use different biological measures to subtype more or less the same sample. Not all measures were there in the whole sample. And then we had a problem that the subtypes, the different clustering methods, were not, let's say, overlapping enough. Then we came to identify the problem that typically, if you subtype, they give you the answer like this individual, this case is subtype 1, 2, or 3. So they give you a binary score. What we did is transform the outcome of the subtyping approach into probability score. So how likely is this to be subtype this and this is the probability. Then by some manipulations on the whole matrix, we were able to harmonize the different subtypes across the different subtyping approaches. And we came from this to this, suggesting that these subtypes and the profile of these subtypes were largely overlapping. And so there we find a way to meta-cluster subtypes derived from different biological measures, which is another way to validate. Now, just one last example from our autism research, where we were focusing on the response to faces for autism. If you present a face and you record the EEG and the ERP, the response to faces, the N170, is slightly longer in those with autism. The latency is longer in autism. It's a minor difference. It's not a way for a diagnostic marker. But still we found this interesting because it would link in with biological hypothesis about autism. What we did is we did check whether, yes, we found it in children, autism longer, adolescents, adults longer. We did check whether it was replicable across the different sites. It did associate with age, but it did not disturb the group differences. It was not a marker of IQ or performance or ADHD comorbidity or other internalizing or externalizing scores. So it was not confounded too much by IQ. It's a relevant issue for autism research and comorbidities. And then we thought about biological validation. So could we link the signal with another biological approach? And so this first approach, the N170, is looking at a computer screen, just at the face. Here we took a task in the same sample, It's called the Hariri task for emotional phase matching. You see this phase and you say, which of the phases below is matching this phase? And this is the shape task, is the control task. This is a task for amygdala activation. And then we found that there was a correlation between the N170 latency and the bold response in the amygdala correlated within the group autism, but also in the typically developing controls, correlated 0.27 with the amygdala response, with a complete different biological task. Then we said, if this is really something which is tapping into the latent biology, pathophysiology of autism, it should also correlate with polygenic risk scores for autism, which it did. So it did correlate with the biological, so the longer latency was associated with higher polygenic risk scores. Then we did check whether it did predict course of autism. So we had 2 waves in our sample, and we took the N170 at baseline, and it proved to be a significant predictor in the violence scores 1.5, 2 years later, controlling for the early violence scores in such a way that those with a longer latency, were less likely to show improvement in cognitive and social and communication development, and those with a shorter latency, more the latency that was comparable to the normal developing individuals, those with a shorter latency were more likely to improve over time. Now, this is an example of a biomarker that we discussed with EMA. And what we concluded with EMA is, okay, if we could enrich clinical samples for those with a longer latency, you are less likely to have autistic participants who are probably improving by the natural course over time. So they could a little bit disturb or confound a long-term treatment intervention study. So if we would enrich them with a longer latency, you have probably a slightly more typical, slightly more homogeneous biological group. And so we check this now in older samples. We check this in a clinical trial, whether it can predict response in a clinical trial. A clinical trial, by the way, to tap into the GABAergic system. So we also test the excitation inhibition hypothesis. And we were able to confirm the N170 that it's likely to be reflecting the biological process, because in higher risk infants for autism, we could identify the precursor for the N170, which was also different, particularly in those high risk infants who switched, who made a transition to clinical autism. So just to show you this as an example of that, yeah, if you start with a biomarker, you try to build a whole confirming network, like different validation approaches, and it gives us overall more confidence that we are really trying to put our finger onto a latent biological process that will keep us busy for many years to bring a hopefully qualified, validated biomarker to the market. I skip this slide because this, in my view, is the research agenda. We should be very critical. What are really the critical, urgent clinical questions, research questions that a biomarker should address? And what is the context of use? We should think about cost-effective biomarkers, because apart from, let's say, the discussions earlier about the MRI is, of course, completely unfeasible in any clinical practice. So even if we would have an MRI biomarker, we should try to translate it into a biological approach, maybe EEG or something else that's more affordable, payable, cheaper, because not any healthcare system can afford paying for MRI for diagnostic assessments. We didn't talk about dimensional disorder models, but is it reproducible across categorical dimensional models? Independent biotypes should be reproducible. Are the subtypes stable also at different stages? Late onset ADHD, adult ADHD, childhood.
Now, just not a short overview about autism biomarkers.